Hi, my name is P.Goodwill, and I'm here to talk to you today about dissemination of magnetic particle imaging technologies. As we get started, I wanted to give you a very quick overview of the entire project that we're working on. So the problem that we're trying to solve is perfusion imaging and cell tracking are underserved by modern diagnostic imaging. And our technology, known as magnetic particle imaging or MPI, really revolutionizes how we can do these two applications. The impact is that we can get better and earlier disease diagnosis for much improved patient outcomes. It addresses a very large market. In the clinical side, it's about 40 billion. And then for MPI specifically, it's about 8 billion. Now, we've had a number of great achievements to date, including a successful preclinical business, which I'll talk about today. We've really proven the technology, and we have controlling IP with about 20 patents. We're peer validated. We've taken in about 6 million in grant funding, and we have growing market adoption. And lastly, we have a clinical prototype under construction, which I'll also show you today. So what's the problem we're trying to solve? Well, we're trying to improve how we do perfusion. Now, perfusion simply is an image that shows how blood flows through an organ or tissue. It's an incredibly common application. There's about $8 billion of scans of this each year, primarily of the heart, followed by the brain. Now, usage is really growing, especially as the population ages. Now, the problem we're solving is modern perfusion has limitations, primarily due to the physics. So on the right here is a CT brain perfusion image. Now, why am I showing a CT? CT is the perfusion market leader. And I'm going to say CT's modern ones are just amazing. This particular CT scanner that took this is the best of the best. It's a 256 slice Philips, got the best of the best reconstruction. It's got like a very high dose. And still we get an SNR of around 5 to 7. We're essentially starting to reach some of the physics limitations of the techniques. Now, here's what you can see. Unfortunately, this patient has a huge stroke, but here's what you can't see. Smaller flow abnormalities, accurate flow numbers. I mean, if we take the same source data and put it into another analysis algorithm, we're going to get a very different result. So MRI and nuclear medicine have different, but similar enough problems. So what does this mean? With current technology, we miss smaller abnormalities. We've got a reduced sensitivity and specificity to what we would like. And since we're using radiation and a contrast agent, we can't really do a lot of repeated scans. Now with our technology, magnetic particle imaging or MPI, we can see exceptionally small quantities of a safe tracer in the blood. So also the tracer itself is safe. It's a rust nanoparticle. So let's see what it lets us do. Here's an actual MPI scan of blood flow. We're seeing a tracer flow into the brain of a rat and then flowing back out. It's got amazing contrast. Essentially, MPI doesn't see anything but the tracer, doesn't see tissue, so the contrast is perfect for that tracer. It's also quantitative. So you can see over the course of 10 minutes, blood flowing in and out. So this enables us to do things that we've struggled to do with medicine. So for example, survey penumbra and smaller vasospasms and stroke, improve reliability of perfusion imaging in general, and can even replace contrast-enhanced CT for cancer diagnosis. We'd still need the CT, but we can do the contrast enhancement. So this has an incredible impact on medicine. We should be able to diagnose diseases of flow earlier and with better sensitivity and specificity, and overall improve patient outcome. So how does our technology work? So the way it works is we use some really large magnets to produce a very unique magnetic field. Essentially, these two magnets are pointed at each other. So essentially, it's the North Pole pointing at the North Pole. And at the very middle of the magnet, you get what we call a field-free region. In this case, it's a line. And whenever that field-free region passes over a nanoparticle, the nanoparticle flips. And we detect those flips with a receiver coil. We take all of those detected flips, put them together, and we're able to produce an image. And in this case, the image is the blood volume of a rat brain. And this is an actual MPI image. The way I like to think about MPI, it's like nuclear medicine with a non-radioactive rust tracer. And these tracers have also been approved for human use in the EU and the US. And so it really leverages the existing technology that we have. And as a side, it's also the first clinically capable medical imaging modality to be introduced in about 40 years, really since the introduction of MRI. So what's our business model? Today, we've made a successful preclinical research business. So the overall goal of the research business is to, one, establish the market, really get MPI into the market, get key opinion leaders on board, prove out the technology. Our customers right now are researchers, essentially PhDs, some MDs, and these guys have been working on applications like inflammation imaging, tracking T cells, perfusion imaging. And then in the preclinical market, our products are instruments, software modules, reagents, and sales and service. So currently we're on track to make about 20 million in revenue by 2025. And then there's the clinical market. This is really the treasure at the end of the rainbow, and it's what we're scaling our technology to. The market is as large as MRI or CT at about 8 billion. And we really don't compete with existing technology. We end up growing the market. Our applications are just different. So our customers are radiologists, and the people that buy the instruments are hospitals and imaging centers. For the cost of the systems, we follow the MRI model here. Instrument costs are about 1.5 to 2.5 million. We expect a service fee of about 10% of the purchase price of the system with a 10-year lifetime. Revenues are an even split between new instruments and sales and service. And if we're as successful as the intro of CT into the market, we can expect about 600 million in a year, 5 years after the clinical launch. So how did we get to where we are? So for the company timeline, our development process has really been driven by funding. So we founded ourselves in 2015 as a spin out from UC Berkeley, which is University of California, Berkeley. And we started actually with a phase 1 SBIR grant. And what that grant enabled me to do was to recruit my UC Berkeley team, essentially the grad students I'd been advising for a number of years, recruit them into the company, and for us to sign up for an incubator. Now, working on our technology led to about 5 million of seed funding, and getting a fully working Momentum prototype, which we installed at Stanford for testing, let us move into our first facility, which is a very small 6,000 square foot facility, and let us launch our Momentum scanner. Now, in about 2018, we started pushing the Momentum beta development and testing, and that was with the phase 2 SBIR, which led to an $18 million Series A led by 5AM Ventures in San Francisco. This let us get Momentum to production. It let us get installs at multiple sites, which you can see over here. And as an aside, this seems like a lot of money, but it's not really. The 18 million has been primarily used to scale the commercial organization and to scale manufacturing. For new product development, we're still heavily relying on NIH funds. NIH funds essentially defray about half the costs, which is primarily the prototype development phase of new product development. For products that we're just coming out with, such as our X-ray CT add-on to the Momentum, which you see over here. We just launched that. Clinical development, which is not shown on this page, and software such as reconstruction tools, imaging analysis tools, the ability to do color imaging in MPI, and a number of other innovative research applications. So let's take a little bit closer look into the preclinical business. I first wanted to share 2 major moments in our preclinical development. First is our install of a prototype system at Stanford. This was, again, developed under a phase 1 SBIR plus seed funding. And here's a picture of me at the install with me for scale. Now, we turned this product into an entire production line. So if you take a look at the bottom here, this is the facility we moved to in 2019, where we continued development of the Momentum system with the phase 2 grant. And this is after the culmination of that Series A led by 5AM. And that Series A let us move into this large facility and staff accordingly. And we really needed both the NIH funding and the VC funding to be able to reach this milestone. And we essentially used the NIH funding to reduce the technical risk to an acceptable level for the VCs and to demonstrate applications. So that NIH funding was incredible here. So what's it take to get into the preclinical market? Well, it's a pretty standard path for any early technology. It's essentially that adoption curve. And right now, we've essentially passed through the innovators phase. We've introduced the first commercial system. That commercial system solves pain points that we couldn't do before, especially for cell tracking. We've also started to see our first pivotal publications. For example, in cell tracking, T cell tracking, inflammation imaging. And we're also starting to see others innovate on our platform. For example, people are developing new nanoparticles, and the nanoparticles are improving the capabilities of MPI substantially. So with that in mind, we're now well into the early adopters phase. Customers are defining applications models, which means they're developing their new animal models specifically for MPI. And they're really helping us develop best use practices. So for example, how do you quantify an MPI? What are the detection limits? Like, how do you label cells better for MPI, et cetera? And this is really giving us credibility. However, as part of the early adopters, we're still waiting on some of these breakthrough publications that our customers are working on. And they're coming out, and they're actually moving along quite well. Coming up is when we'll be getting into the early majority, and that's where we're going to need significantly more publications, and we're working with our customers to make that happen. And that's also when our clinical translation has to start to get on the horizon, and something's got to be there. Otherwise, the early majority, they're all banking on us being able to get to the clinic in the near future. So the preclinical market adoption is definitely accelerating. I wanted to show you guys some applications that we've seen over the last year. So on the top left, we see CAR T cell tracking from the University of Florida, perfusion imaging from Stanford, smart drug release monitoring from Michigan State. Essentially, we can tell when doxorubicin is being released, but we do this magnetically, and we can see this throughout the animal. There's no death limitations. Robarts Research is tracking circulating tumor cells homing to a cancer. And these are just a small snippet of the different applications coming out. If we take a look at the number of MPI applications and publications across the literature, we see that there's just been this exponential increase, and we don't see any sign of it plateauing. And the press is also noticing. We've had some really nice publications highlighting MPI in the non-academic press. They're essentially from our customers over at Stanford, UNSW in Australia, Robarts in Canada, Michigan State. And we even had a recent article in Forbes. So how do we get into the clinical market? So in 2015 to 2020, I talked about how we made our first steps to establish the MPI market via small animal research. And this is mice and rat research. So as we go into 2021, this year and 2022, our major goals for the clinical market are to finish designing and building the clinical scale prototype. Prototype means it's just, first attempt at it. And we're well underway and have quite a few of the parts in. Right now, we're targeting neuroimaging, and we're starting with neuroimaging because the brain doesn't move, unlike the heart. And that's very helpful for technology development. We're also going to be able to do large animal testing if we're able to raise the next round of funding. And this also lets us begin design of the clinical beta. The clinical beta is essentially the instrument that we want to clinically introduce, which is separate from the prototype. Prototype is a technology demo. A beta is what, the shape and form that we want to do introduction of. So in 2023 to 2024, we're going to begin building and refining that beta system, and we'll seek an IDE exemption to test on humans. And last, we'll be able to start human testing. After 2025, that's when we can perform clinical trials. After trials, we're aiming for a 510K de novo. For the 510K de novo, we're targeting an orphan pediatric disease, and that would let us qualify for fast track exemptions. So where are we right now on the clinical program? So let's start with the program milestones. The first thing that we're doing is we're working with the NIH via the CARE program to approach the FDA. We're pretty early on the clinical program, and having a program like this, where we can talk to the FDA in a way that doesn't bind us as a sub-Q, is actually quite helpful, and it's something that we're looking forward to. Unfortunately, for the overall program, we've had some delays due to COVID, but we anticipate we'll finish most of our milestones. On the technical side, we've now got a prototype under construction, and we're even getting some of the parts in. So shown on the right, we just put in our shield room, and there's a bore hole over here for the instrument. And also the main bore of the system came in. And I'm holding a screwdriver next to it for scale. It's a pretty large bore, and it can fit the shoulders, the head, the whole body of the patient. So to summarize some of our key accomplishments, we now have a proven technology. We've actually got multiple preclinical products. We've got controlling IP and around 20 patents and growing market adoption. You can see the map up here on the top, and the orange dots are one of our hardware instruments. That's our Momentum MPI imager, and the blue dots are another one of our hardware instruments that we sell into the preclinical market. On the clinical side, our prototype is under construction and it's moving quickly. So I want to introduce our board of directors. On the top left is Anna Christensen, our CEO, followed by David Allison, a partner at 5AM, Victor Wang, a partner at AIMTOP, myself, I'm the CTO, and Joe Victor, who's the CEO at Raresight. We've got an amazing advisory board, Steve Connolly, who's also a co-founder of the company. He's over at UC Berkeley. Max Wittermark, he's at Stanford Neuroradiology, and he's helped setting the technical direction of the clinical scanner. And Mike Harsh, he's the ex-CTO at GE, who's helping us understand some of the business implications of our clinical scanner. We've also got some wonderful investors, 5AM, who ran our Series A round, and AIMTOP, Sandhill, and Stardex, who did our seed round. So with that, I wanted to say that this work was not done in a vacuum and relied on the amazing help of our collaborators at UC Berkeley, University of Florida, MSU, Robarts, and Stanford. I'd also like to thank the staff of my company and, of course, the NIH for funding. So thank you very much, and I'd love to take some questions.